Cool, so is it... You can hear me? It's going? Okay. Cool, well, thanks for the opportunity. So, kind of recent scientific developments from genetics to microscopy are really opening new avenues in terms of the research and experiments that we can do in terms of understanding biology. And kind of in particular interest of me is the question, how do organisms develop through the movement and division of individual cells through space and time? And so one tool that's particularly useful for this, as Joe was kind of just mentioning, is light sheet microscopy. So just a quick kind of speedy tour. And so what light sheet microscopy allows us to do is image an individual plane that leads to a 2D image. And so here we can see this is a developing zebrafish. And the idea is simply just by stepping through the volume, we can stack these together and create these 3D images in a 3D volume. We can then do this through time and create movies. And so this here is a 3D movie through time, tracking the cells of a developing zebrafish. And so color here is just the depth. And so kind of these rich data sets, could potentially provide the basis of a really quantitative and data-driven approach to understand development really at the level of individual cells. And so if we're going to really do this data-driven approach, we don't really just want to be able to analyze one 4D movie. We really want to be able to do this across multiple N samples, where N could be 10 to 100. And so in many areas, we're kind of at the area where the experimental techniques and the microscopes are really there to start generating the data. It's just the terabytes of images that we end up with create issues in terms of even just storage, visualization, and then eventually the segmentation tracking or downstream processing that we really want to do. And so, first thing that comes to mind is just what about image compression? And so as you previously saw, these images are rather spatially sparse. And so what compression techniques allow us to do, as we know, is to bundle up our data set to something small. And this really helps us with things such as storage and data transfer. But when we actually want to go back and do processing tasks on this data, we basically just have to blow it back up into our original data set. And so although this kind of helps address issues such as storage, the idea would be, could we not do something that basically helps out the whole entire pipeline, that allows us to utilize the sparsity to help out with both storage, visualization, segmentation, tracking, everything kind of downstream from acquisition. And so that was exactly the idea behind my PhD project, and resulted in the development of what we call the adaptive particle representation. So, a quick aside is, if we then thought about the visual system, and the visual system does a similar task, we take in photons, and we're processing and making decisions. And so what if our visual system worked the same way as a microscope? And so that we fix the spatial, the temporal resolution, and then also it was in color, and then our eyesight can see about 32 bits. Sorry.
Could you talk a little bit more about the physics of the light-matter interaction? I mean, is it just attenuation?
So exactly how this imaging processing works.
A little bit more. Is it membrane? Is it cytoplasm?
Okay, so the scale that I'm looking at, so these here were nuclei that are labeled. So the general idea is kind of any contrast labeled structure. So where they've been labeled with fluorescent proteins, and then they basically, a light is shining on it, it illuminates and emits another wavelength, and then you detect that.
So it's pre-emitted?
Yeah. Sorry. And so if this was the case for kind of the human visual system, we'll be producing something like 1,000 gigabytes per second, or this is about 90,000 terabytes per day. And obviously, this is not the case. And so research has shown that, or estimated that the eye actually is only, through the optic nerve, is only transmitting about 1 megabytes per second. And so what are two features kind of visual processing? One is that there's adaptive sampling. And so we all know this, that our fovea is constantly kind of adaptively sampling the world. The second is this features kind of a local gain control. We know that as we turn the lights on and off, we're basically able to still realize that there's a mug in front of us. And so absolute intensity is not really what drives kind of the visual system. And so the idea is, so instead of, we have our pixel images, which are rather sparse here and in, where they're evenly sampled. The idea behind the APR is simply just have an adaptively spatially sampled representation, where on the left here we just, particles are just like pixels that can be adaptive in size. And the idea then is that by focusing our particles onto the information content, we can then focus our memory, our storage, and our processing tasks downstream. Kind of the upper logic of this approach is that if we take two different points here, these stars, is effectively that we want to constrain the absolute error between what we can reconstruct from this image and was in the original image by some user set bound E, where we have this sigma here. And so what is this? So it's this sigma we call a local intensity scale, and we can think of this as kind of an analog of this local gain control for the eye. And so why do we need this? Because in fluorescence images, you can have the same structure in different parts of the image that have completely different absolute levels of intensity. They can be they could be 10 to 100 times different intensity. And if we have a technique that isn't aware of this, we'll end up resolving potentially bright areas well and under-resolving dim areas. And so simply by taking this into account, we can then resolve both kind of dark and light areas simultaneously. So going a bit more into the tools and mechanics of actually how the APR works. And so it has two components. One we call kind of a resolution function, which is continuous, piecewise continuous across the domain, and another set of particles which actually store our function. And so as I said, what we want to do is find the pair of these such that this reconstruction condition here holds for all original pixel locations in the image, where this reconstruction is any weighted average of particles within r of y distance of y. So if we choose y, r of y says how big of a kind of resolution or spatial neighborhood we can use. And so this is just basically then just a weighted sum over images. And so here just having a look at a simple Gaussian, so here you can see that we have our adaptively sampled set of particles and our associated resolution function that goes with it. And so what is the actual kind of optimization problem that we're solving here? The idea is that we want to effectively find the largest possible ever neighborhood, r of y, such that this constraint is satisfied. And so it turns out that solving the discretized form of this is n squared complexity. And so even for small kind of images, this becomes really unfeasible. So instead, kind of the classic mathematical approach is we try and solve another problem which is simpler that we can. And so it turns out that we can actually solve, we focus on the second bound, which comes from a Taylor series expansion. And so it turns out that if this bound here, which relates this resolution function to the gradient of the image, is satisfied, then this reconstruction condition is also satisfied. But what you'll see here is that this isn't easy either. This is also an n squared complexity problem, and it has the r of y on both sides of the inequality. And so it turns out, though, that if we restrict the functional form of our r of y, we can then solve this problem in linear complexity, and actually becomes really quite fast. And so what are kind of these restrictions? And so what it ends up coming down to is you end up tiling your domain with kind of Lego pieces of different sizes, where they're restricted to be powers of 2, and where both this tiling tells us about the size of the resolution function at each point in space. So for example, anywhere in this box, the resolution function will have a radius or a size that is equal to the size of that square, and similarly then for a smaller box. And it turns out then that we have this, then what do we do with the particles? Well, since we've got a nice simple representation of the resolution function, we can basically just place our particles directly in the center. So how do we go about producing one of these APRs? So imagine you can start with a fluorescence image. You select your user threshold. I don't know whether you can see that E there. You basically compute your local intensity scale, which is essentially just a local standard deviation estimate. Then compute the gradient magnitude, kind of combine them together, the ratio, and then you solve them using this linear algorithm, which we call the pilling scheme, to basically get the solution here. And then we can directly just sample our image, and we have our representation. And so, of course, this is not just in 1D or 2D slices. It also works in 3D. And so here, what you can see here, I don't know whether with the light you can see it properly at all. So this is a 3D video just showing how, in red is basically pixel resolution, and yellow is going down to the lower sampling, that this also kind of is homing in on the content in 3D. So what you'll probably be able to see better is this example. So if we then go back to this 3D image here of Felucia, this is the original image, and what you're now about to see is exactly this resolution function, where what white will basically mean pixel resolution and progressively darker means lower resolution. And so you can see that it's just capturing basically the information content of the underlying image. And so just then quickly, here on the left is the original image, on the right is a reconstruction. And so this is kind of, because the edges are at pixel resolution, you don't really see much of a difference there. But the fundamental idea is that the APR adapts to the image contents while guaranteeing a user set reconstruction error, E. And so then the idea is that this adaptation focuses storage, computation, and memory on the important areas. And so to give you an idea of kind of what impact this can have on fluorescence images, so we took 19 different examples of light sheet microscopy data with kind of a mean average size of about 1.8 gigabytes. And so in terms of just the compression task, sure, these are quite sparse. We can achieve nice compression with an average of about 100 or a median about 40. But then when we go in terms of the number of particles versus pixels, so the number of computational elements you have to process over, you can see that then also we have a reduction of about 50. And then further, it's not just the computational elements, it's also the required amount of memory in your computer when you're actually programming over. And so this, then you can see that about 37 times less. And so kind of this idea of adaptive sampling is not new. And so in image processing, there's kind of a lot of techniques that go into a similar direction for things like superpixels, adaptive mesh representations, wavelet thresholding, or pyramid schemes. And actually the area that was kind of more influential from the motivation of my ideas here was actually in the area of differential equations, where adaptive numerical schemes have found a lot of use. But the kind of the overarching idea behind the APR was to be as simple as possible, such that the algorithms could be easy to implement and as close as possible to the original pixel algorithms that exist. And so one of the problems is that kind of the task here is that we want processing directly on the APR to accelerate and enhance existing approaches, rather than replacing it, more that you have your current processes, you have your current algorithms, and that you can just re-implement them directly on top of this. And so to kind of go deeper in this, processing on the APR, you can think of it just as kind of pixels of different sizes or volumes. An alternative things that also sits well, it has a multi-resolution pyramid, where you can just think of it as a truncated tree where you're just storing things at the leaves. It also naturally has a graph representation, so if your algorithm works over a graph, you could do it there. And finally, if your algorithm requires kind of a local isotropic patch, you can also easily do this on the fly. And so in terms of data structures, then what do you kind of require here? Well, if we take this example of the 2D APR, basically you can see this is just a breakdown of sparse matrices. And so for each resolution level, you effectively have a sparse matrix. And for almost all the algorithms that I've developed, the only kind of thing you need to be able to do is iterate over a non-sparse direction, usually without random access. And so what I'll just go through is kind of some image processing examples of these different representations. And first off, for the graph representation. And so one kind of classic segmentation approach is that of graph cuts. And it's well known that this kind of is memory hungry. And so across the test image data sets, I don't know whether you can see this, would take about 400 gigabytes on average to process these data sets. However, we can directly use the APR graph, and it's only 13.5 gigabytes. And so actually I could process all of them on my laptop here. And furthermore, we also get a speed up that's directly proportional to the pixels and particles. And so here's just an example of segmentation and a direct rendering of fish vasculature of the head. Another kind of task we have is actually compression. And so rather than being actually a compression technique in itself, it can be used in addition to existing approaches. And so, for example, there was recently a compression approach known as within noise level compression. So the idea is you can actually take the APR and further apply that. And so here what I'm going to show you for the traditional, on the original image, we have some parameter that effectively we decrease the image quality and we get better compression ratios. And you can do the same thing directly actually on the APR, and the only difference is you get a similar trade-off. You basically just get more bang for your buck. And of course then visualization, and so one nice thing is actually you get kind of additional means, and so here this is just directly rendering actually the particles, and you can see you get this kind of new really spatial tactile view of your sample. But of course you can also do traditional approaches such as ray casting or maximum projections straight on the data. And so then you have the savings of the original data was about 13 gigabytes, whereas the APR on disk is about 24 megabytes, or in memory is only about 54 megabytes. And so of course, you all know here that the power of deep neural networks and machine learning, and of course if we want to have the APR as being our main kind of representation of the data, we need to be able to perform our convolutional neural networks or neural networks directly on the representation. And so now kind of what we require if we want to do a convolutional neural network, we effectively need two things. We need a pooling operation and we need the convolution. And so here's this just another one way of thinking about the APR. So we have the pixel image and its associated kind of downsampled tree. And here you can just see the APR. And it has an identical kind of structure. We're just truncated basically the tree at certain places. Sorry.
Just to check, with respect to when you have temporally related images, are there temporal restrictions on?
Sure, so I'll come to that later. So kind of at the moment I'm discussing just spatial adaptation, and then we have an approach of kind of doing it through time that I'll come to in a second.
Kind of lifetime.
Yeah, exactly. So first off, just the pooling. So we can see that pooling operations are natural, so there's not much that's need to be done there. But then we consider convolution operations, we see that we have a problem here, right? So we've got a convolution where we no longer have particles at that resolution level, and of course on the fly we can just reconstruct this with a piecewise constant reconstruction. And actually, we can see from this data structure that it's natural to allow our kernels to depend on the resolution. So where we have potentially different stencils that depend on the local resolution level that you have there. And so this is then work that was done by a talented master's student, Joey Johnson. And so currently we just have classification tasks working. But so here the simple task that we tested against is we had kind of 6 different types of objects where they were a triangle and a fuzzy triangle, kind of circle, fuzzy circle, square, fuzzy square. So we wanted the algorithm to have to be able to basically capture these high resolution content. And so here we actually see that yes, these APR nets can do the challenge, and actually it seems that they possibly have a kind of higher capacity than the equivalent kind of pixel net, or just maybe easier to train. And so if we have a look at a bigger net, we see that the pixel net's in blue and the APR net's in green. So we're seeing that it indeed can work. And beyond the synthetic example for natural images, here you can see we have this, we did an old Kaggle challenge, which was cats versus dogs. And so here you can just see the impact of the selection of this E parameter on the representation here. And so for all these results, we chose E equals 0.2. And so again, you can see that the APR net also is successful. And again, it seems to be kind of training a bit faster. So what would be the benefits? So the idea is that you can get reduced memory and computational cost through doing this. And second is that it seems to regularize the training process by focusing learning. And so this is an interesting area, but we've still got to do a lot of work. So we've still got to add the upsampling GPU implementation and really start actually kind of doing some ablation experiments on understanding why we're seeing kind of these improvements in the learning rate. And so kind of all of this comes together in software. And so what we have is what we call the LibAPR, which is a C++ library. And so the idea here is that it implements both OpenMP and CUDA implementations for transforming images into the APR. And the idea is that we kind of implement basic image processing building blocks. And the idea is that then through wrappers, we can interact with kind of existing software. And so the main focus at the moment is both Java and Python. And so, for example, for the machine learning project through PyBind 11, we managed to basically interface with PyTorch to get the machine learning working. And then with Fiji, which is an image processing software that's really big in the biosphere, we're kind of getting things going there using Java CPP. However, this is definitely a work in progress, trying to really make these tools useful. So as was kind of previously mentioned, I've talked a lot about space, but what about time? And so it turns out that we can kind of extend this, our reconstruction criteria, where we not only have kind of some error that we're willing to make in space, if we allow ourselves also to make some error in time, we can then do a similar thing where if we just have a look here, we've got some moving data set, and kind of things slow down, less objects are moving, and then again, we have it starting up again, where this top blue line is just the traditional APR, so it's unaware of whether things are moving or not. And then the second is the APR plus time, which is then exactly accounting for these different dynamics in time. And so the idea here is that we wanted the APR for each time step to still look the same. And so we kind of do a spatial APR first and then do this time adaptation kind of on the fly afterwards. And so, for example, here the original time series was 6.7 gigabytes. The APR of it was about 150 megabytes, and then the time version is about 45 megabytes. And so actually beyond just this classic here, so beyond just doing reconstruction bounds on our function f, so here I've dropped the sigma for ease of notation. We're solving problems like this in linear time. We actually can solve kind of a general class of problems where we can then bound the different derivatives of our data set. And then furthermore, we can actually kind of combine these together simply by just taking the minimum over these functions. And so this kind of provides this toolbox of adaptation that you can apply to potentially a range of different problems. And so one kind of application area is kind of going full back. I had inspiration from numerical differential equations, is to go, can we then use this for differential equations? So in my research group in Dresden, we have one of our core competencies is for large-scale simulations, distributed simulations on meshes and particles. And so here you have potentially sparse in space data sets that can take up to weeks on high-performance computers to solve. The idea is, can we then use the adaptive particle representation as an adaptive technique for these numerical simulations? And so this is work in progress, and hopefully the idea is to get both GPU and distributed versions of this code going. Yeah. And so kind of in my mind, where I like to think of my research is going from this really large spatial temporal experimental data and trying to develop techniques to go further and further, reduce it down further towards biological insight. And so you can really think that the APR is right at the beginning of that chain of taking the spatial data and kind of reducing it down and focusing on important areas. Another area that I'm kind of interested in is taking this experimental data and reducing it down further into models, for example, partial differential equations. So there's been work recently on where we have spatial temporal data, and from this we want to directly learn what are the partial differential equations that kind of could describe that process. And so, for example, we could have concentration fields, for example here. This is, they could come from some reaction process. And the idea is that from this we want to directly learn basically the PDE governing equations. In this case, it's the Gray-Scott model. And so kind of there's been two kind of really recent work here. And so I'll just quickly go into how this kind of works. And so imagine what we have is a discrete sampling of our data. So this is in 1D. We have it in time and just in space. And so the idea is that we basically propose that the underlying data follows a PDE of the following form. So we have both linear and nonlinear terms that we're trying to discover, but we don't know what they are. And we assume they're sparse. And so then basically the idea is we set up the following minimization problem, where we first then compute the spatial, the temporal derivative directly from the data set, and then compare this to the possible complete set of our spatial derivatives, which we again estimate directly from the data set using techniques such as finite differences. So the idea then is that you could use regression, or what's shown to work better is sparse techniques So for example, one of the pioneering papers showed that lasso or L1 regularization can work pretty well. And further paper came out that showed that they find that actually this idea of sequential threshold ridge regression works better, which is a combination of L0 and L2 norms. But to me, there's still challenges here of actually, showing it on synthetic data where you know the outcome. What about, actually being able to apply this to real data? And to me, there's kind of two challenges here, is that these regularization models depend on parameters that are sensitive to the noise level, the sampling, and the dictionary size. And so, although you may be able to show you can recover these models, they often depend on basically setting your parameters correctly. A second thing is that the results are sensitive to your choice of method of derivative estimation. And so there, you had to compute your temporal and your spatial derivatives. And if you have noisy data there, this can be a real issue. And so how are you going to do things there? And so I've been lucky to be working with Christian Müller, a data scientist at Flatiron Institute, and so Madhu, a PhD student, on kind of trying to tackle these two problems. And so in terms of the first problem, we've worked on using some existing recent techniques called stability criteria for kind of removing the dependence of these parameters across different types of sparsity models. But the second thing that I want to kind of talk to you today about in detail is addressing this problem of the differential estimating of derivatives. And so this is what I call kind of the coefficient moments approach, where instead we basically regress directly on the function or the data values here. And instead then we basically on a stencil that's centered around the point but excluding the middle point. And so here you can see that you have combinations of both just the stencil values, just like a convolution, and you then also have this nonlinear term here, which basically combines. So we have a linear stencil, and then we have nonlinear stencil, which is basically pairs of points that we have to select. And so the idea is that we regress against and we find alpha and beta, and then you can actually compute A directly from the alpha and beta that you get in your data, which is, so we can go back and exactly do our original task of predicting that model. Kind of, how does it work? Just kind of a brief sketch. So if we have a look at the linear case at least, we know that if we have some kind of local stencil which approximates our differential operator with some convergence rate, we know that that must basically satisfy this relation here, which is depends on what's called a Vandermonde matrix, which is essentially just monomials of basically the points in our stencil. And so the idea here is that by simply we estimate now instead our coefficients, and we can basically then through multiplication say what differential operators they are approximating. And so what are the potential benefits here? And so the idea is that you can learn a data-dependent differential operators that kind of adapt to the noise level, the sampling rate, and of your problem. Second is it's parameter-free. It actually kind of allows for a more general class of models in terms of your spatial derivatives, your temporal derivatives. And furthermore, you can actually use this framework for estimating coefficients of known models. So let's go into a bit more detail of a given example here. So for example, here we have data that's taken from a viscous-Burgers equation with certain parameters. So here's space and then evolution through time. And so what we can then do is first have a look at the noise-free approach. And so here I'm not using a sparse regression, I'm just using classic linear regression, and I repeat it 100 times. And so what we can do is then have a look at the coefficients estimated and kind of get an idea of the stability through kind of a t-statistic type thing. And so here we see that the direct approach works reasonably well. And then we also then have a look at the coefficient approach, and we see again that it's working. And actually what's interesting is we can see that we actually get the coefficients down to machine accuracy here. And then we can also actually have a look at the learned kernel. So what are we actually learning from the data? And it's actually quite surprising that this looks so scattered. And you're seeing that actually you have very large coefficients really quite far away from the initial point. The real difference then also becomes once you change and increase your noise level. And so here in this direct approach, you see that although it's capturing the first nonlinear term, here which should have a coefficient of -1, it loses basically our second derivative completely. However, then when we have a look at the coefficient approach, we find that we still retain the two terms being basically the most significant. And a nice property that I find across kind of experiments here is that if it finds something and it's significant across all the repetitions, it's usually the true coefficients. So when it kind of fails, it just finds nothing, which is quite nice. And so if we have a look at the learned kernel here, we actually see something completely different, where it's actually really the sparse kind of spread out, more blurred kernel, which makes sense given it's a noisy environment. And so then the second thing that I was saying that you could do, so suppose that we now know that we have this Viscous-Burgers equation and we just want to estimate our parameters from our data. And so you can, of course, then just regress your computed derivatives and terms and you get some estimates. But it turns out that actually this coefficient approach is actually really good at estimating kind of known model parameters from the data here. So it gets a really nice estimate there. And so kind of what I've shown is that, from discrete samples of continuous spatial temporal data, we can extract these partial differential equations. But one of the kind of questions for me now that I'm really interested is, imagine now we have our cell tracking data through time, and we have across samples and potentially something to do with the state, whether expression levels. Can we do some sort of similar approaches where we can learn models and integrate it across data sets? And so there is kind of in the first question of actually what are the kind of models that we want to be learning. And so with that, I just want to thank all the people involved in this research in the labs and questions.